All right, so I'm going to talk about BANKSY algorithm for spatial omics, integrating or analyzing various kinds of spatial omics data to infer various properties of the tissue. And first, as you've gotten a flavor from the meeting so far, in spatial omics, sure, technologies are moving very, very fast, but data analysis is a major challenge. Okay? So the algorithms have not kept pace, and definitely benchmarking has not kept pace. So what we want to do is take data sets that look like this. So in this case, this is the dentate gyrus of the hippocampus. You have many cell types in the spatial arrangement. You want to be able to take this data and say, okay, what are the cell types? What are the domains in my tissue? Now, the problem is that, visually, you can see a lot of patterns, right? Because the eye is tuned very nicely. The human eye is tuned for looking at spatial patterns. But bioinformatically, people are still, to a large extent, using single-cell RNA-seq algorithms for cell clustering, which ignores spatial information altogether. And that's quite, I would say, a statement on either people's awareness of spatially informed algorithms or the state of the field of development of spatially informed algorithms. Either way, it's clear that there is information in the spatial arrangement of cells, right? So if we want to identify tissue domains, I want to identify cell type clusters, we should be able to get better results if we incorporate spatial information intelligently. And I won't get into why I said the word, intelligently or appropriately. There are some methods being developed right now, which are quite sophisticated for incorporating spatial information into cell type clustering, but they either introduce unexpected biases or they're extremely slow and non-scalable. So what we want is a spatially informed algorithm that's very, very fast and scalable, because as you've seen, you can generate data on hundreds of thousands of cells and millions of cells, spatial data fairly quickly. So you want a super scalable algorithm, not something that stops at 10,000 or 20,000 cells. And that algorithm should take both the cell's transcriptome and some spatial information into the clustering. And that was the goal, or that was the starting point for our method. So if you go back to this image, you can see here that each cell type has both a characteristic transcriptome, right? It expresses certain genes represented by specific colors. And you can see here that, for example, white cells are surrounded by white cells and a little bit purple cells. Green cells are surrounded by green cells, and they're also in the neighborhood. There are purple cells and orange cells and white cells, and so on. Right here, you have a layer of orange cells mixed with purple cells, and so on. So each cell has not only a characteristic transcriptome, which is what the traditional clustering algorithms look at, but also it has a characteristic neighborhood. And so why don't you use both of those kinds of information for the clustering? And that's what the BANKSY algorithm does. So BANKSY stands for building aggregates using a neighborhood kernel and a spatial yardstick. And BANKSY's philosophy is that a cell is known by the company it keeps. What does that mean mathematically? So you're all familiar with the gene cell matrix that you use for clustering, right? Each column here is a cell, each row is a gene, and this purple rectangle is the expression level of all of these genes in all of these cells. Now, BANKSY does something very simple to extend this. BANKSY says each cell is represented not only by its own transcriptome here, across, let's say, 5,000 genes or 500 genes or whatever, it's also represented by the average transcriptome of its neighborhood, okay? So let's say this cell is this cell. This cell's own transcriptome is inserted here in the expression matrix. And the average transcriptome of these cells is inserted here right below it. So now the cell is represented by an augmented feature vector consisting of own transcriptome, neighbor transcriptome, multiplied by these scaling factors, parameterized by just one free parameter, lambda. You can see here that when lambda becomes really large, you're mainly weighting the neighbor transcriptome. When lambda is very small, you're mainly weighting the own transcriptome. Okay? And the distance between any two cells, if you work out like this, the square distance between any two cells is 1 minus lambda times the own expression square distance plus lambda times the neighborhood square distance. Right? And you can tune lambda and give different weights to the own expression and the neighbor expression. Now, the beauty of this approach is that you just change the feature space. You don't have to change anything else. And now you can throw this clustering algorithm into ScanPy or Seurat, or this data matrix into ScanPy or Seurat or anything else. And those algorithms will cluster this. They'll think this is single cell RNA-seq data. They won't know this is spatial omics data. And they'll cluster it using the standard approach. And you'll get sensible results. And because these standard approaches are highly scalable, BANKSY is highly scalable. So I should say this is largely the work of Vipul and Nigel, two very talented postdocs at GIS, along with key collaborators, Kok-Ha Chen, who was one of the creators of the MERFISH technology, and Li Hui-Kwan from the Bioinformatics Institute in Singapore. And please check out our bioarchive preprint if you want further details on this. So this is the logic of BANKSY. You can think of it as it's just one simple neighbor augmentation step of changing the data matrix, and after that, everything stays the same. Okay? It's very elementary. So you can use your favorite clustering algorithm, or you can use your favorite feature selection algorithm. You don't have to change anything else. So what is the intuition behind it? You can say, let's say the x-axis is the cell's expression, okay, some metagene of the cell's expression. The y-axis is some neighborhood expression metagene, okay? Now you can see here these blue cells and the green cells. If you look at only the cell's own expression, they kind of intermingle. You know, the blue cells and the green cells, they are separated. There's some ambiguous cells. You don't know whether they're blue or they're green. But because they have different neighborhoods, their neighbor transcriptome is very different. And so in the neighbor transcriptome space, the blue cells and green cells are far apart. So then you can draw this dividing line and very cleanly separate them. Right? And that's the advantage of this neighbor-informed clustering. And you can change lambda. If lambda is 0, then of course you're ignoring the y-axis. You're multiplying the y-axis by 0, and it's just traditional single-cell RNA-seq clustering. If lambda is very large, then you're stretching this y-axis, and you're clustering mainly by the neighbor expression. We use some intermediate values of lambda typically for spatial cell type clustering, or typically lambda of 0.3, where you get a balance of own expression and neighbor expression, both contributing to your clustering. So how does this work in practice? Here's an example, mouse hypothalamus, 3D spatial data, and this is from the Moffett et al. 2018 mouse brain paper, MERFISH. So you can see here, this is one section of the brain. Visually, you can already see there's one zone here and there's multiple zones here. Now, if you do traditional non-spatial clustering, you get mature oligodendrocytes all over the place, okay, in both zones. But if you use BANKSY, the mature oligodendrocytes split cleanly into two different clusters, the dark red cluster and the orange cluster. They have different spatial domains and different tissue architecture. Now the question is this an artifact of BANKSY? Is this real? Are these genuinely different cell types or subtypes? So here are those cell types again. One cell type based on non-spatial, two subtypes based on BANKSY. Now here you can see what happened. First, look at just the upper half. This is the gene cell matrix. These are some marker genes of these two cell types. Orange means these cells. Dark red means these cells. You can see here, if you have the right color scale, you have the right marker genes, you can see in this heat map that yes, these are two distinct cell types or subtypes. Only, but subtly distinct. Okay, there's very little expression difference there, even though it's systematically different. But when you look at the neighbor transcriptomes, they are sharply different. And so the neighbor transcriptome really helps you highlight this subtle difference, which becomes a strong difference when you look at the neighbors. And so I won't show it here, but we took these genes. Now, because of BANKSY, we know the markers of these two different subtypes. We went back to single-cell RNA-seq data and said, if you take these markers and cluster the cells, will you identify two distinct cell states? And indeed, these sets of markers, they give you two distinct cell states in single-cell RNA-seq that correspond to these two different subtypes of mature oligodendrocytes. So we looked at mature oligodendrocytes, same thing for excitatory neurons. With traditional non-spatial clustering, excitatory neurons all cluster together. With BANKSY, you get two different subtypes of excitatory neurons with two different spatial zones. Again, you can see here the own expressions are a bit clearer. Okay, there are genes that clearly separate these two cell types, but only a handful of genes. And that's why traditional single-cell clustering could not identify it. If you turn up the resolution of traditional single-cell clustering, you'll find this in a slightly fuzzy way. But you'll find a lot of other artifactual clusters that are not really cell states. So that is one demonstration. Let's look at a few more case studies. Now, this is MERFISH data. Let's look at SlideSeq data. For SlideSeq data, people use deconvolution methods because one spot in SlideSeq can actually cover two cells sometimes, or 2.5 cells, and so on. Right? So people use methods like RCTD, which is a deconvolution method. It's more complicated because you need a reference single-cell RNA-seq data set with cell type labels and so on. But RCTD in the brain, you can identify, say, a granule cell layer, okay, in the SlideSeq data when you have some reference single-cell RNA-seq data. So we said, can we use BANKSY to analyze the same SlideSeq data set without giving it any reference, without giving it any information? Will it be able to recover the granule layer and other layers? And indeed, if you look at the top row, these are two different SlideSeq data sets. BANKSY beautifully recapitulates these different layers and cell types in the mouse brain. Very similar to what RCTD gives you, but RCTD needed external information of labels, single-cell RNA-seq data. BANKSY doesn't need any of that external information. And in fact, you could argue the molecular layer into neurons, BANKSY catches them even more cleanly than RCTD, if you look at. And non-spatial clustering also identifies these layers, but not as cleanly. And it's more noisy, particularly the granular layer. And then here are the marker genes to corroborate the same thing. So I've shown you two data types, MERFISH and SlideSeq, where one was analyzed using clustering, one was analyzed using deconvolution. BANKSY can replace both of those approaches. Then case study 3, VeraFIS data, which is also a multiplex fish-based assay. And again, mouse brain. Here you can see non-spatial clustering. Each of these non-spatial cell type clusters was split into two different subtypes by BANKSY. And these two different subtypes had very sharply different spatial distributions. And we're comparing not just to non-spatial clustering, but also to Meringue, which is one published spatial clustering algorithm, and then to Giotto, which is really another spatial clustering algorithm, but for identifying tissue domains rather than cell types. Neither Giotto nor Meringue. So Giotto finds one of these spatial subtypes, okay? But it mixes these two together, the CA3 and somatry sensory cortex cell types. It mixes them together. It creates some strange clumps in the hippocampal area thalamic nuclei. It creates some strange clumps here because Giotto forces cells that are next to each other to be the same cell type, or encourages them to be the same cell type. And that's because Giotto was not designed for cell type clustering. It was designed for domain segmentation. Meringue was designed for cell type clustering, but in practice, we find that Meringue results look very much like non-spatial clustering results. So BANKSY is the only algorithm on this data set that could identify all of these cell types cleanly. We did not have StereoSeq data at that time, but we hope to incorporate StereoSeq data as well. And BANKSY, because it's a very elementary general purpose method, it should apply to StereoSeq data just as well. And here are the markers showing that the clusters that BANKSY identifies are not random. Right? There are specific cell type markers of each of these cell subtypes and they are known in the literature. You can look at them in situ hybridization images as well. Then one more case study. We've talked about cell types. We talked about in base data types. If you use a very high value of lambda, I mentioned that BANKSY mainly pays attention to the neighborhood expression. It does not pay attention much to the cell's own expression. And when you do that, BANKSY becomes a spatial zone finding method. So in this case, we use BANKSY to find zones in this brain organoid data generated by my collaborator, Jinye Liu at Genome Institute of Singapore. These are the brain organoids. These are the total gene counts. You can see here that in the center of these organoids, right, you're not detecting many genes. Why is that? Because the center of the organoid is necrotic. But still, there's some cells near the periphery of the outer edge of the center. They passed our QC, but BANKSY in zone finding mode with high lambda said that these cells just on the inner core, they form a cluster, the red cells, in multiple organoids. And these are essentially necrotic cells. You can see here, they express heat shock, upregulate heat shock proteins, and they don't express clearly any other cell type markers. And they are spatially where you would expect the necrotic cells to be, near the core. Right? And they have kind of marginal detection of genes. And then BANKSY also detected this very interesting green cell type, which is a misdifferentiated cell type, which is expressing markers. This is supposed to be four-brain organoid, and these are not four-brain organoid markers. So this is a misdifferentiated cluster that, again, we discarded. So we discarded red cells, we discarded green cells, and then did our downstream analysis to infer the biology. So you can see here that in zone finding mode, BANKSY can do QC, cell type QC, as well as in clustering mode, it can identify cell types. Lastly, I mentioned BANKSY is super scalable. The y-axis is the runtime, blue is conventional clustering, red is BANKSY, conventional single cell RMC clustering. Okay, let's say you're in Surat or something. BANKSY runtime is almost identical to that, as you can imagine, because BANKSY is just a feature augmentation step followed by conventional clustering. This conventional single cell clustering scales easily to over 1 million cells. We are in the process of benchmarking runtime for over 1 million cells. I don't have the latest update yet. But these other algorithms at around 30,000 cells, they stopped. And at least on the hardware we had, they were not able to cluster any larger data sets. And 30,000 is quite small. You want to get to 100,000 million cells for a spatial omics algorithm. Lastly, benchmarking on Visium cortical layers. Again, so this is a popular benchmark used in the field. Visium data, one more data type. This is the ground truth, okay? Manually annotated cortical layers in this brain section. Here's the BANKSY result, and here are the results from other algorithms that have been tested on this. And you can see here, BANKSY is the last algorithm. It gives you the best correspondence to the manual annotation ground truth. So just one single algorithm, optimizing lambda, you can analyze so many different spatial omics data types, get robust results really fast, identify subtypes, identify tissue domains. And so we're very excited about BANKSY's applicability. I mentioned my co-authors. Thanks also to Rima from the group and Genome, which kindly supplied the VeraFIS data to us. And A* for funding. Thank you.